Hi class, Dr. Jim here. Welcome to the first lecture of microbiology this semester. In this chapter we're going to be looking at a number of different things. Really it's a good introduction to what we're going to be focusing in on all semester long. And this kind of gives you an idea of the different things we're going to look at, some of the history behind microbiology, and kind of some of the ways that we classify the microorganisms. So it kind of gives you a really good indication of what's going to happen throughout this semester. So let's begin with our lecture here, and we'll go through the slides as we go along. You can follow along, you can take notes, and figure out what you need to do in order to be successful in this class. So let's begin. So let's start. So first we're going to find out what is microbiology. So what we're going to do is actually go through the process and talk a little bit about what is microbiology. You may have taken biology before with me or with someone else and known that we talked about a lot of different things in biology, but this is going to focus in on the small things, the very, very small things that you tend to see with a microscope. The second thing we're going to look at is the history behind microbiology. So we're going to talk about a few different things. First, we're going to talk about spontaneous generation. This is the idea that things magically appeared out of thin air, that basically that inanimate objects gave rise to animate objects, meaning that dead things could then become alive and make new live things. And so we'll talk about that. We'll discuss a little bit about the germ theory. The germ theory is the idea that germs actually cause disease. Now, before the late 1800s, many people thought it was God's way of punishing people, and maybe some people still do, is that disease is spread through God's will. But this is obviously not the case. Through modern medicine and modern microbiology, we know that this is not the case, and that microorganisms are the ones that are responsible for causing disease. And then finally, we're going to look at two main characters that really changed the face of microbiology. So first we're going to talk about Louie Pasteur. You probably have heard his last name used quite a bit if you've ever drinking a gallon of milk or orange juice or maybe even some beer, and that's called pasteurization. This is a method he developed in order to kill microorganisms. The other guy is Robert Koch. Robert Koch was a figure at the late 1800s, early 1900s who really established the germ theory and saying that microorganisms were responsible for the different types of diseases. And so we're going to take a look at what some of his accomplishments, what he's done throughout the course of history. These are the two main guys that I really want you to know. We'll talk about a few other figures in microbiology, but really Louie Pasteur and Robert Koch are the two main guys that I really want you to focus in on, and I'll ask questions about it on the next test. The last thing we're going to look at is how do we classify microorganisms. And so this is going to be an important process in which we look at how we establish the names, how we name things in microbiology. It's very similar to what we see in biology, but microbiology, we're talking about the small things, bacteria, viruses, sometimes eukaryotic pathogens and things like this, but typically things we can't see without a microscope. Okay, so let's move on. All right, so microbiology, what is it? Well, microbiology is the study of organisms that are too small to be seen without magnification. And again, we have a lot of different microorganisms that are included in this group. Probably the one that you'll be most aware of is bacteria. Now, bacteria is what we're going to be doing a lot of in the lab. And so when you come into lab every week and that stuff, we're going to be working primarily with the bacteria. And again, these are going to be very small organisms. They're called prokaryotes. We'll discuss what those are in the next chapter, and we get into chapter 4 and looking at the difference between prokaryotes and eukaryotes. But bacteria are these really small, tiny cells, very basic types of cells. We'll also look at viruses. So when we get into chapter 6, we'll talk a little bit about viruses. Viruses are not really a cell. They're called infectious particles, and because they are made up of protein and nucleic acid, they're very simplistic. They're even more simple than the bacteria and much smaller. And so we'll talk a little bit about those, and again in chapter 6. We'll also discuss a little bit about the fungi, protozoa, helminths, and algae. These are all eukaryotic pathogens. And again, some of these things are small, like the protozoa. These tend to be single-cell eukaryotic cells, and again, you want to look at them under the microscope. If you looked at pond water, you'd see these things swimming by and doing these things. But most of these other things are actually pretty large. If you're talking about worms and people getting worm infections and things like this can be really big. And we'll talk about this in chapter 5, some of these guys, the fungus, the protozoa, the algae, and the worms themselves. And again, these are going to be sometimes multicellular, have really good developed organ systems and things like this. And we'll look at this in chapter 5. Okay? So that's where we're headed in the next couple of lectures. All right, so what are some of the endeavors in microbiology? So if we look at some of these different things, we can see that a lot of things came off the basic microbiology. If we think about microbiology being the study of very small things, we've developed a lot of techniques that have been utilized using these very small organisms. One of them is the study of your immune system, and this is called immunology, and so this is going to be really important. And again, this is one of those things that we really want to focus in on. We get up to chapters 13 through 16, and we're going to look at these different things and how your body actually fights infection. Another thing we'll look at is, again, public health and epidemiology. This is the tracking of diseases and looking at who's getting infected with what, how are they getting infected, and why are they getting infected. And so it's kind of the study and tracking of diseases. If you're interested in a lot of mystery type novels and where did it come from and who did it and why they did it, this might be the topic that you really want to get into. You can actually go to like UWM and go to the school. So at the main campus, the UWM campus is right next to it, that has the School of Public Health, and they actually have an epidemiology program in which you can take once you get into either undergraduate or graduate school and earn a degree in that and get your public health degree. And so that's basically tracking the diseases. We also look at biotechnology. Biotechnology is a huge field, lots of jobs and lots of different things in the area of that, and again, can be very popular. We do offer classes in biotechnology. These are taught by Barb Wimpy. Very well established, a lot of research behind what she's done. And so if you're interested in these types of classes, I highly recommend looking into the program and seeing maybe this might be a profession that you might be into. Along with this, the genetic engineering and the recombinant DNA biotechnology. And if you look at this, this is again the manipulation of genetics using either genes from one organism and putting to another, or modifying the genes. And this is what we call these genetically modified organisms. So if you've ever looked at a package that you might buy at the store, a lot of times they'll say either GMO free or GMO, or some indication of what a GMO is. What a GMO is something that's been modified in the laboratory. The genes have been modified to change it or add genes to something to give it more nutrition or other things like this. It's highly controversial in the sense that some people think you're making Frankenstein and changing things, and these things now get into humans and that stuff. There are other people say that it's perfectly safe. And we'll look into that when we get into the genes and genetics in chapter 9. But again, this is something to kind of think about. And again, being educated is what I want you to get out of this. Figuring out what it is. And then you make the decision on whether you want to actually eat it or not. That's up to you. Okay? So those are some of these microbiology things that have progressed out of the beginning of microbiology. Okay, so where did everything start? Okay, and so we look at this little timetable up here and we look at what are the origins of the microorganisms. And so if you look at the idea that the age of the Earth is roughly around 15 billion years old, maybe a little bit older than that, 16 billion, and it all depends on what people think geologically, or in the idea of space-time and things like this. Well, for the longest period of time, for about 10 billion years, there was really no life on this planet. It took time to cool the planet down, to actually get rock, solid rock, actually form water and other things like this. And until you had the right combination of ingredients, did we not see any cells. And the first cells we saw were the prokaryotes. And we're going to talk about the prokaryotes again in chapter 4, looking at these. These are very simple cells that have some DNA on the inside. They have a membrane surrounding it, and that's about it. And then they have some fun stuff on the outside that we'll look at again in chapter 4. These guys ruled the world for about 2 billion years. And so from 4 billion to 2 billion, these guys were the only things around. Eventually what we saw is more complexity that occurred. This complexity led to what we believe is the eukaryotic cells. Eukaryotic cells, the difference between that and prokaryotes is eukaryotic cells have a nucleus. They have a nucleus, they have some organelles and things like this that do some of the processes. We start to see multicellularity here. We start to see more and more of the diversity of plants and animals, and finally we get up to where humans are. Now, one of the things I like to discuss is where do we all fit in this biological world, or in the time. So if you think about time as a clock on the wall, if you look at the clock on the wall and where do we fit into that, think of the Earth starting at 12 o'clock midnight, let's say on Sunday night. Okay, so at 12 midnight, that's when the Earth started. Okay, if you go all the way around through 12 o'clock the next day, so 12 noon on Monday, and go all the way around to about 6 p.m. Monday night, that's when the first cells were actually appeared. So it took a very long time for the first cells to even show up. Okay, when we think about where humans end up on that, we have to go all the way to 11:59 and 59 seconds on that clock. So right before the clock strikes midnight, 1 second before, that's where humans appeared. So if you look at it, humans have a very small portion of that clock. Okay, evolution has taken a long time to go from the prokaryotic cells, these single cell organisms, all the way up to humans. And that 6 hours that it took to get through, now it doesn't seem like much, 6 hours, but that's a very long time in the idea. So you're talking about 4 billion years before humans and anything else started roaming the planet. So kind of gives you some perspective on how long things are actually happening. Okay, so again, we're going to look at two types of cells. And again, in chapter 4, we'll talk about the prokaryotes. These are microscopic, unicellular. They're always found in unicellular. There's no multicellularity. And again, lacking nuclei or membrane-bound organelles. The other types of cells we're going to be looking at are eukaryotes. So when we get to chapter 5, we're going to talk about the eukaryotic system, those types of cells. You're going to see some are unicellular, but many are multicellular. When you talk about the more complex animals and plants and things like this, these are going to be multicellular. And again, they have organelles that do a lot of the functions inside cells. In chapter 6, we're going to look at viruses. And many of you you've probably heard of a virus, but you probably really haven't studied much about viruses before. Maybe in A&P, you may have heard of some things like this, but probably generally you have not. And so we'll examine what a virus is. And again, we don't consider them cells. We actually consider them as infectious particles. They have protein on the outside and nucleic acid on the inside. And so we'll discuss this again in chapter 6 and kind of give you an idea of what these are. These viruses are much, much smaller than what we think of bacteria, and really, really tiny compared to eukaryotic cells. So we're going to see a very big size difference when we look at these different things when we get into the next couple of chapters. All right, so this kind of just gives you an idea of all the different types of things we're going to be looking at throughout the semester. So again, most of the stuff we're going to be looking at is going to be the bacteria. This is Mycobacterium tuberculosis. This is the bacteria that actually causes tuberculosis. And so again, you see these different shapes and things like this. This is called a bacillus, or rod-shaped. And we're going to talk about this again in chapter 4, so don't worry about scrambling and writing this down or anything like this. But this just gives you an idea of what the types of things we're going to see. This is a fungi. A fungi is multicellular. It's eukaryotic. And again, people have fungal infections a lot of times. We call these mycoses, where people get these fungal infections. So things like athlete's foot or ringworm or some of these other things. These are types of fungal infections people get. Algae tends to be not a parasite on humans, but it can cause problems with toxic particles and releasing toxic agents into water and things like this. And so some of you may have heard of Toledo, Ohio, a couple of years ago, where they actually weren't able to drink the water, and that was because of these algae blooms. And again, that's something that we worry about because it poisons the water. You can't drink it, kills all the fish and a lot of different things, and we're seeing these happen more and more often. And again, this is due to a lot of things that actually are happening due to man dumping phosphates and other things into the wastewater, and it's getting in there. So we'll again discuss that more as we get into the algae. Some other things, this is the virus. These are very tiny particles, and so this is the herpes simplex virus here. These are the ones that cause cold sores, and so you may have seen people, or you yourself have suffered through a cold sore, and this is a little opening in the skin. It's an infection of the subcutaneous skin in that, and it causes an eruption that occurs for a couple days and then goes away. And again, we'll discuss more about what a virus is here in a couple days and look at those different things in the next chapter, or in chapter 6. Another guy is the protozoa. Lots of different types of protozoa over there. These are like animal cells, but on a small scale. So we're talking unicellular. They have to eat, and they do have a tendency to have parasites associated with them. And so we'll discuss this. We get into a later chapter, chapter 23 and 24, when we're going to be looking at parasites and those things. And one of the main parasites are protozoa, where they get in and cause infections and things like this. And then the last group that we'll be looking at again later on in the chapter, in chapter 23, are the worms. And so we call these things helminths. That's the Latin word for worm. And again, this describes the different types of things. Here you see cyst of a roundworm that causes trichinella. This is when you don't cook your pork chops long enough and that stuff, and you can end up getting these types of infections. This worm actually goes and cysts into the muscle. It leaves the intestine, goes into the muscle, and can cause problems. And we're seeing this more and more in the southwest United States from meat that's imported from Central America and Mexico, and people are getting these infections due to eating undercooked or raw meats in this case. And again, we'll discuss more about this in chapters 23 and 24. All right, so this just gives you an idea of the sizes of what we see. And so most of the things that we're going to be looking at are very, very small, okay, in the range of micrometers to nanometers, so very, very tiny. So if we think about what a meter is a big meter stick. And again, we'll look at this in the lab. We'll talk a little bit about the sizes and things like this. When we work with microscopes, I'll go through a little spiel about how we do conversions and things like this and look at these things. But think of a meter stick, pretty long. It's about 3 feet. And that, when we get into the millimeters and centimeters, we're talking about the really small things. You can barely see those. We're now talking on a magnitude of 1,000 times smaller than 1 millimeter, and that is a micrometer or a micrometer. And so very, very small, 10 to the minus 6. You may have seen this before in biology. Maybe you've talked about it a little bit in A&P, but this is a very small number. And then when we get into the viruses and that, we're now talking about the nanometer range, which is really tiny. This is now about 1,000,000 times smaller than a millimeter. So we're talking very, very small, or 10 to the minus 9. And so you can kind of see the ideas of how big or how small these things are. So we're talking about bugs, and these are tiny bugs like a louse or a lice type of thing. This is 1 millimeter in size, so very small. We get into the bacteria. A lot of the bacteria are pretty tiny. These are anywhere from 1 to 10 micrometers in size. So the E. coli or Staphylococcus are very tiny. And then finally, when we get into the viruses, these are very, very small. These are about 1,000 times smaller than the average bacteria, so very, very small. And then you get down into the atom range in that, and then you're talking about very, very tiny in this case. And so we'll look at all these different things again as we go through the chapters in the next couple of days. Okay, so one of the things I like to do is throw some concept questions in as you go through. Now, I will read the question and give you the answers, and I want you to think about a little bit. You might want to pause it here and think if you kind of test yourself. I will give you the answer after a couple of seconds, and then we'll move on. So one of the things we're asking is, which of the following things does not describe a fungus? Now, I didn't talk too much about a fungus here in this case, but I wanted you to kind of get an idea of what some of these things are. So, does a fungus contain a nucleus? Does it have ADS ribosomes? Is it useful in decomposition, or is it photosynthetic? Now, again, this might be a question that if you didn't have biology before, it might be really tough. But one of the things that we know about a fungus is fungus do not have chloroplasts. So again, they do not or undergo photosynthesis or photosynthetic. And so this one's kind of a tough question. Sorry to start off a little rough here and that stuff. It'll get better as we go along. But I like to throw these in just to see if you're paying attention. Not worth any points or anything like this, just to see if you're paying attention to the lecture. So we'll go on. All right. So how do humans use microbes? So when we think of microbiology, a lot of times what we think of is disease, infection, what happens to people when they get sick. Well, there's a whole other realm of microbiology in which we utilize microorganisms to our advantage. And so some of these things that we talk about, and again, we'll bring them up later on in the semester when we talk about DNA and some proteins and things like this. But one of the things that are very useful is biotechnology. And again, we see things now. We can manufacture foods and drugs and vaccines using these microorganisms. It's a very important part of the field. And like I said, if you're interested in doing some of these things, and we're going to try and do a few of these things in the lab this semester, But if you want to really hunker down and do some cool things, you can always look at the biotech program that we have and take some of the classes that we have. Very, very popular classes, some of those things that I think they're really good techniques and things like this that you can really learn and learn something from. Okay. Another thing is genetic engineering, and again, this is changing or moving genes from one organism to another. That's the GMOs that I was talking about before. Again, very important. And then the last thing is bioremediation. That's something that we haven't really discussed. Now, this is actually where you use microorganisms to clean up different waste or different spills or things like this. So probably the most common place where you hear it is like when we have oil spills that occur out in the oceans and things like this. So some of these companies now actually harvest bacteria that can eat up oil. And so what they do is basically dump the bacteria into the water, and they would go and eat up the oil slicks so that the oil can escape, and then the oil is degraded by the bacteria and the site is cleaned. And so these are some of these new technologies that are coming out that are really important, and a lot of people are getting into these fields because there's a lot of money to be made. So if we can do this naturally, the way that the earth is actually doing it, instead of using harsh chemicals or harsh conditions and things like this, bioremediation is a nice way to go and try and do things naturally. Okay? All right. So what are some of the lifestyles of microorganisms? And so this is where we think about, where do microorganisms live? Most of the microorganisms that we ever come in contact with are free living, meaning that they don't care, they don't want to be on you, they don't want to have anything to do with you. They're pretty much free living. We see these guys in the air, the soil, the water, on surfaces, everywhere. And again, they really don't want to cause you harm. They don't want to even be bothered by you. And again, it's one of those things that they're just the free living organisms. Most are harmless, and again, some can be very beneficial depending on where they live. The ones obviously that we worry about and why you're taking this class is the ones that cause disease. And so we determine these guys, or we describe these guys as parasites. These are the ones that live on or in body of another organism and can cause damage to the host. And so this is the thing we worry about when we talk about infections and things like this. So probably the most ones that you're going to hear about this semester as I describe these different microorganisms are the ones that cause disease. But I do want you to know that there are plenty of microorganisms out there, far more microorganisms out there that don't do anything to you or actually provide a benefit to you. So don't think that a germ is a bad thing all the time. Sometimes these things are very good, and maybe some of you take them, called probiotics. So we'll see. We'll talk about that later on as we go along. Okay, so when we look at diseases, obviously when we think of diseases, we term these things as pathogens. Pathogens are microbes that cause or do harm. Okay, there are roughly about 2,000 different microorganisms that actually cause disease, and you can see on the side over here on the right side, oops, I'm sorry, how many different diseases are actually out there. And again, this is just when we're talking about diseases and things like this, is not every disease that is out there caused by microorganisms, but for the most part, these are the main diseases that we see that are caused by infectious particles. Roughly 10 billion new infections every year worldwide. And again, you think about the population, we have about 7 billion people. That means you roughly suffer from 1.3 infections every year. And so you probably get sick probably maybe once, maybe twice a year. And that's about average for most people. Sometimes if your immune system's weakened, or maybe you have a little one at home that goes to daycare and they bring everything home with them, you might get sick more often. Other times, some people don't get sick at all for a year, and good for you. You got a really healthy immune system if that's the case. But roughly, most people suffer from 1 or 2 infections at least a year. And so, are those numbers going up? It depends on who you ask and what you look at and things like this. But that's a whole nother story for another day. Okay. And again, there's about 12 million deaths from infections worldwide, and so this is obviously a big number, so people do die. And so again, we'll be looking at a number of different diseases when we get into the upper chapters from chapter 18 on, of things that are trying to kill you and how they kill you and that stuff, and we'll discuss how fatal these things are. But you can see over here on the right, we have a lot of different diseases that are caused. By far the one that causes most problems is the respiratory infections, and this includes pneumonia and influenza. Flu kills more people every year than a lot of the diseases that we suffer from. And so it's one of those things that you just say, oh, I got the flu, and then you live and you go on and no big deal. But flu kills a lot of people every year, and you don't hear much about it. You hear, you hear it in the news, oh, there's an outbreak of flu and that stuff. But when we hear Ebola, everyone panics because, oh my God, we're all going to die. And really only about 2 people, in the United States got it last year, or maybe 4 people got it, and only one of them died from it. Okay, and so you look at this, flu is by far more pathogenic, one that causes more disease and causes more deaths per year than any time ever Ebola has. Flu has killed more people every year than Ebola has ever killed people. So think about that the next time when you talk about flu or Ebola or freak out about different things. Okay. Another big disease, obviously, is AIDS. Obviously, HIV, we'll discuss that as we go along throughout the semester. Diarrheal diseases, obviously, when you have a problem of dirty water, or drinking dirty water, or contaminated food, you always have a problem with this. And then your simple bouts of food poisoning and things like this can always cause problems. Tuberculosis, even though in this country it's not a big major disease, it is in many other parts of the world. And so, again, we have this free-flowing society that go from place to place. So again, not to say that you're going to catch tuberculosis or anything, but it is very common outside the United States. Other diseases, malaria. Malaria is probably by far the worst parasitic disease, and I'm talking eukaryotic parasite in the world. Kills more people, kills millions of people every year. Roughly about 300 million cases every year of malaria, and that equates to about 3 to 5 million actually dying from the disease. And typically small children are the ones that are most at risk for that disease. We also talk about measles. Measles, again, not in this country, although when we have outbreaks like we did last year or 2 years ago with Disney World or Disneyland, where we had the outbreak, and that tends to be for people that are not vaccinated. So if you've been vaccinated with the MMR vaccine, you're protected. You shouldn't have measles anytime in your life. Hepatitis B, a lot of us have to get vaccinated, especially if you're going on to be nurses or other healthcare professionals. You have to be vaccinated for hepatitis B. It's another one. Again, it's a bloodborne infection that people carry and tend to carry, but now that we have vaccines, less likely. Tetanus, parasitic diseases, and other things out there are, kind of wrap it up. But by far the biggest disease that is out there is the respiratory infections. And again, this includes colds, pneumonia, flu, things like this. And again, most of us suffer at least once a year with some type of infection in this state as we go on. Okay, and if you look at looking at diseases in the United States versus the world, you can see there's only 2 infectious diseases in the chart in the United States. And again, very low numbers. I mean, 65,000, that's a lot of people that die from that. And again, 33,000 die from that. It's quite a huge number. But again, when you compare that to heart disease or cancer, it's far less. It's, only a tenth of that, of what you see either for heart disease and cancer. And so it's one of those things, yeah, obviously we're concerned with, but it's not something that we're all paranoid about, oh, are we going to catch this? You go into other parts of the world, and you can see respiratory infections is the 3rd leading cause of death. Infections, and this thing, HIV and AIDS, diarrheal disease, tuberculosis, malaria. You don't see these things in the United States because the United States is more developed. Our healthcare system works pretty well. I guess that can be up for debate. We get vaccinated for things, we're protected from things. Other places in the world, not so lucky. And so we see a lot of these diseases. And so I want you to be aware that, yes, the United States doesn't see a lot of infectious diseases, number 7 and number 10 on the list, but you go outside the United States and simple things like respiratory infections like the flu kill numbers of people, diarrheal disease because of the lack of clean water, things like this, tuberculosis, malaria, are huge, huge problems every year that cause death in a lot of people. Okay? All right, so let's move on to the history. So let's talk a little bit about where microbiology came about. And so really, microbiology stems from very early on. So it's kind of the idea or looking at the unknown. And then before we had microscopes, we really didn't know anything about microorganisms because we had no way of looking at them. Once we saw what these things look like, we started studying them more and more, and obviously we saw a lot of these different things. So some of the things that we're going to see today is microscopy was obviously a big thing. We'll talk a little bit about the scientific method and how we perceive things or how we go about testing things. We'll talk about the development of medical microbiology. Why is this important? Why do you wash your hands when you're operating on people? Things like this. And then finally, microbiology techniques. So we're going to look at all these different discoveries as we go about throughout this section. Now, obviously, we couldn't look at microorganisms without a microscope. And so Antony van Leeuwenhoek is the one guy that actually came up with the microscope. And his was a very simple microscope. This is basically a magnifying lens. And so he came up with this idea, hey, I can look at things. And what he did was he looked at things inside his body. So he looked at the scrapings on his teeth. I'm sure he looked at the things crawling in his skin, because people didn't shower, probably stuff crawling in his hair and all kinds of different things. And he noticed that these things were very, very small. Again, he could magnify things up to about 300 times. Our microscopes go up to about anywhere from 400 to 600 times. And if you're talking about oil, up to 1,000 times. So it really hasn't changed that much from Van Leeuwenhoek's time period to now. But again, it allows us to see a lot of different things, and we can see most of the bacteria and other things. We can't see viruses, but a lot of the really small things we can see using the microscope. Okay, here's a picture of Van Leeuwenhoek's microscope, and again, you can see very simple. It was kind of a magnifying lens here. You put the specimen on this little nail here. You can move it up and down. You can move it back and forth to focus it and kind of look at things. And these are some of the things that he actually saw. So he saw some rod-shaped bacteria. He saw some things moving. He kind of showed them through motility. Some of the other shapes that he saw, some bigger things, and again, he was able to describe some things based on the shapes that he sees. And so again, lots of different shapes. And again, we're going to talk about the shapes of bacteria and what they look like here in chapter 4. But again, this is kind of some of the similar things that he saw very early on with this simple microscope. Okay. The next thing was the scientific method, and so this came about at around the same time that microbiology was getting started. And so again, this is the process of kind of figuring out something that you see. Okay, and so again, we basically come up with a question. So when we do the scientific method, the idea is that you're going to look at something for a little while, and then you're going to formulate questions based on what you see. So you make some observations, and then you come up with a question or a guess. And then this is what we call a hypothesis. And a hypothesis is an explanation that you're either going to prove or disprove based on the data you collect. Okay, and so that's the idea. Then what you do is you come up and you come through and make some experiments. You kind of test your question. Is it worthwhile or isn't it worthwhile? You then can publish your data and that stuff. It gets repeated, and then you come up with these different things. You say yay or nay to your hypothesis, and if it is good, you say okay, you publish your work and say, hey, I found something to this interesting question. If it doesn't work, you start all over and you ask new questions. And so you kind of come up with these different things. And that's pretty much the life of a scientist. Okay? If things are well supported, we start to see these things building up. And so this is where multiple people see the same things and can repeat these experiments over and over again. When they're highly repeatable and actually able to be done, we call these things a theory. And so you may have heard things such as the theory of relativity, or the theory of natural selection, or maybe the cell theory, that all things come from cells. And so these are things that are well established. These are ideas that have been well established and been looked at over and over again and been proven to be true over and over again. Okay? And if you get something that's really concrete, very solid, something like the law of gravity, that's when we talk about something becoming a law. Basically, you can't disprove it. So if I drop something right now, like my pencil over here, I grab a pencil and I drop it, I know there's gravity. I can't dispute that gravity doesn't pull that down closer to the center of the earth. And so that is why we call those things a law. Okay, so something that can't be disputed is a law. A theory is 99.999% true, but there could always be that one thing that throws the theory off. And so we can't say that it's a law concrete for sure 100%, but it pretty much is. You know, it's up for debate occasionally, but it pretty much is. And so that's what separates a theory out from what a law or a principle is. All right? All right. So here, concept check. Scientific theory has little or no evidence that supports it and can best be described as a best guess. Is that true or false? Okay. If you answered false, you are correct, because again, a theory is something that gets proven over and over and over again to be very concrete. I just got done saying it's basically 99.99999% true. There could be that one thing out there that throws the whole thing off, but we still haven't found it yet. And so a theory is highly backed, very has lots of good evidence behind it to keep it going. Okay? Good job if you got it right. All right. So the next thing we looked at is spontaneous generation. And so again, this was the idea that things just magically appear. And so spontaneous generation was the idea, like if you think of, if I had a bunch of rags sitting in the corner of my house and I just left those rags there, that all of a sudden that one day some magical force would come down and create mice. Or if I left out a piece of meat on the desk and I just let it sit there, and after a while, voila, flies would appear. Okay, and that's saying that these non-living things would generate living things. And that was the idea of spontaneous generation, that things just magically appeared. There was some life force out there that would zap something that was non-living and make it alive. Okay, well, this was a strong theory at the time of Louie Pasteur. But Louis Pasteur didn't agree with this, and he said, you know what, guys, I think you're wrong on this idea. And so what he did was he came up with an experiment to look at and say, you know what, I don't think you're correct, guys, and I think there's a different way of looking at these things. And basically he went through and he proved that even when exposed to air, that microorganisms are the ones that cause this generation or things to become spoiled, or looking at these different things. And that was using these gooseneck flasks. And I have a video here in the next page that you guys can click on so that you can actually watch it. I'm not going to show it here in this video, but I have it included in the set of videos with the first lecture. And so I highly recommend you look at it. It kind of talks about the gooseneck flask and what he does. But essentially what he did was he disproved the idea that it just naturally happened, that some bolt of lightning came in and zapped it and became a living thing. What it was is that he showed that as soon as that air with the microorganisms in that air landed in that broth, essentially you'd get the life coming from there. So the life was actually in living things that were in the air. Okay? And he came up with the theory of biogenesis, which is the idea that living things only arise from other living things. So you either need microbes or something else that's alive to give you live things. Okay? Now, this is, like I said, I have this video included with you in the packet of videos. It's only like a 2-minute video, so I highly recommend watching this. It just kind of gives you an idea a little bit more of the experiment behind it. It's really good, and again, very short idea of what he did to actually do it, looking at the gooseneck flask. Okay, so highly recommend watching that video. Okay. Some other ideas now, when we talk about the germ theory, the idea of the germ theory is that germs give rise to disease. And so the idea of this was established through a number of different doctors and scientists during this time period after around Louis Pasteur, and that the human body is the source of infection. Okay, so the first one that saw anything was Dr. Oliver Wendell Holmes, and that he observed that birth mothers at home were actually less likely to get infections than those that went to the hospital. And again, it was some of the practices that the doctors were doing at the time that actually established infections, and so that was one of the things that we saw. Another one is the Semmelweis. Dr. Semmelweis showed that, again, physicians were the ones that were actually carrying the disease to the mothers. And so they go from the autopsy room back to delivering babies, and now these mothers would get sick and they would die from it. And again, it was due to the infections that they were spreading with their dirty fingers. And so that was one of the things. The last thing was Joseph Lister. And again, you probably recognize his last name when you see it. Lister, Listerine. He basically came up with solvents that would actually wash the hands clean of infections. And so these are some of those things. We call these things antiseptics. Things that you can put on your hands, your body, things like this that kill germs, that allow you to now go and do different things without spreading germs from one person to another. And so he was the first one to introduce these different things. And so we give Listerine, Lister, a kind of a name from him back from the past of one of these things, again, using these things. And so again, two of the things that he came up with is using disinfection for hands or antiseptics. And then the other thing is using heat to actually sterilize the tools, so that when tools are used from patient to patient, you should sterilize them first, either with heat or, you know, basically heating them up hot enough to kill the germs. And so that's one of the things we obviously do now in medicine to stop the spread of germs. Okay, Again, another wonderful video for you guys to look at. This is called the Germ Theory of Disease. Kind of just goes through again with some nice pictures and things like this. Again, very short video, about 5 minutes long. I think it's a discovery video. Has Bill Nye the Science Guy. Very fun video to watch. Highly recommend watching this again. It kind of goes through again some of the different aspects of the germ disease with this, or germ theory of disease. All right. So when we talk about the germ theory, this is that many diseases are caused by the growth of microbes and not due to being a bad person or by God punishing you, okay? And the two main contributors to this, besides the men that I mentioned before, are Louie Pasteur and Robert Koch. And again, these guys did a lot to the different realms of science. So if we look at these different things of what these guys did, and these are some of the things that I want you to know about these two different guys, okay? So Louis Pasteur, he was the first one. Again, he lived in the 1800s. He showed that microorganisms were responsible for fermentation. So he worked for the wine industry, wine and beer industry, and showed that yeast were the ones that were responsible and not bacteria. And that bacteria and microbes also cause spoilage, and that you keep things open to the air, they're going to spoil and taste bad because the microbes that are in the air land in there and start causing things to fall apart. Another thing that he showed is that, again, the spontaneous generation. So I highly recommend watching that video with the gooseneck flasks and things like this and taking a look at those things. But he showed that spontaneous generation does not occur, that you actually need live things to cause, to give rise to live things. He developed a process called pasteurization. This is basically heating things up to kill the microorganisms that are in the liquid or in the fluid first, and then cooling it back down so that it's safe to use in the human body. He also developed the first rabies and anthrax vaccines out there. And so, again, very important guy. He noticed that people that were exposed to these things over and over again didn't get sick. And so he formulated ideas that maybe if we take these things from these people and inject it into other people, that they might be protected as well. And so, very smart guy for a time when things weren't really developed at the time. So Louis Pasteur, very important for microbiology. Okay, I have a nice little video again, short. This is like 2 minutes long. If you want to take a look at some of the highlights of his career and what he did, some of the different things. It's a kind of a catchy tune, and you'll probably be singing the rest of the time. So again, watch the video, very short. If you have some time to look at the Louis Pasteur video as well, kind of just talks about the same things that I discussed here just a minute ago. Okay. The other guy that I want you to know is Robert Koch. And Robert Koch established the Koch's postulates. And so these are one of those things of basically going through it and figuring out what causes disease. This is the experimental steps to you to figure out what is causing you to kill your patient, essentially. So the steps themselves are involved is the first thing that occurs is that first you see someone come in with an infection. Obviously. So the patient comes in with the infection. That infection, what you do is then you take a sample from that patient and you try and isolate it. That's the second step. You isolate the infection. Okay, what causes the infection? The third step you do is then you take that isolate and you inject it into a live animal, typically a mouse. We typically don't inject it into other humans or things like this, and we look to see, does the mouse get the same infection that that person does? And if they do, and that they get sick from it and potentially die, we say, okay, that's a cause and effect. And then what we try and do is the fourth step, which is re-isolate that organism out of the dead animal or the animal that's sick from that organism. And if we can do that, we've established that this organism causes this disease, okay? And so using those postulates, we can see that there's a lot of different things that actually can be occurred by microorganisms. And again, this is how we identify different diseases, and it's still in use today. Okay. A couple other things that he did was he identified the cause of anthrax, TB, and cholera. So he found the different things using his postulates to figure out some of the diseases that people were getting. So anthrax, TB, and cholera were caused by these things. And then he also developed the pure culture method. So you can blame Robert Koch for making you do microbiology lab. So the Petri dishes and some of these other things that we're going to be using in the laboratory throughout the semester all stems from Robert Koch's work. And so you can blame him for making you have to do all these different things in microbiology. But again, establishing the things that we see today and establishing health and vitality and things like this. We live a lot longer because these guys figured out certain things that would kill us throughout our lifetimes. Okay. And again, I have a nice little video here about Robert Koch. It's again a short, I think it's about 2 or 3 minutes. If you want to learn out a little bit more about the different things and how we look through these different discoveries and that stuff, highly recommend. I talked about it already. Not saying you have to watch it, but I highly recommend it. Take a couple minutes, and I think it will give you a good idea of what these guys actually did. All right, so the last thing we're going to discuss in this lecture, and I promise I'm done and I will shut up for today, is we're going to look at taxonomy. Okay, and taxonomy is how we organize these things. So organizing, classifying, naming things in biology. It's a chapter of biology in which we organize things. As you know, scientists are very anal. They like to organize things. They like to have these things. If you're very anal and like to organize things, then maybe taxonomy is for you. This was a formal system that was originated by Carl von Linné. He was the guy that came up with the idea of giving organisms two names. Okay, and so it concerns with classification. How do things fit together? We talk about the common ancestor. How do things like humans relate to bacteria, or humans relate to dogs, or whatever you want to talk about in these situations? And we look at these things and we want to know how these things are classified. So what we do is we assign names based on what they look like, some of the different molecular things that we see, and we can identify them through a lot of different techniques that we use in order to look at these things. The identification is giving them a name so that that name can be used to identify these things wherever you go in the world. And once you identify those things, you can figure out what you have, okay, and where they place within that chart of all living things. Okay, so this is a level of classifications, and again, most of the time we're just concerned with the domain. And so in this class, we're just going to talk about domains, being the archaea, the bacteria, and eukarya. And again, we're going to talk about this in chapter 4 and 5, so I won't spend too much time on this right now. But again, we'll look at domains throughout the course of the semester. Kingdom is associated with the eukaryotic organisms, and so you know your kingdoms of plants, animals, fungi, and protists, and those are the kingdoms. We get into phylum and those, but really the ones that I want you to focus in on along with the domains is the genus and species. That's basically the first and the last name of an organism, and so we'll talk about this. You have a first and last name, so do all organisms using genus and species. And again, we'll talk a little bit about how you use genus and species in identifying an organism here. Okay? So if we look at the domains, again, the domains, in fact, are looking at the types of cells that we talk about. We have two types of cells. We have the bacteria cells, and we have the archaea cells. And these are two different types. And again, these are both prokaryotic cells. And again, we'll look at these in chapter 4. And then by far the bigger class of organisms are the eukarya. The eukarya are the larger cells. They have the nucleus, they have the organelles, they do the different things, and so those are going to be important. We call these the eukarya. These have the nucleus, they have the organelles, and again, we're going to look at these in chapter 5. The plants, the fungi, the animals, and the protists are the ones that we talk about with kingdoms. And again, we won't really discuss too much about kingdoms because we're not too concerned with the eukaryotes. Most of our class time is going to spend with these guys down here. But again, we'll look at these over and over throughout the semester. So we'll take a look at these things. Okay. So again, here's the different domains. We're going to be talking about bacteria and archaea. And we talk about how everything is related. If we look back, we have one common ancestor, the first cell ever on the planet, and then everything branched off of there. And this is the idea. And so we'll talk about these things again in the various chapters as we go on. So bacteria, archaea, and then eukarya. These are both prokaryotes. These are eukaryotes. These are the bacteria that infect us. These are the ones that live in extreme environments. These are the ones that make up us. Okay, and so we'll look at these different things as well. All right, and so here's some sample taxonomy. And again, I'm not going to spend too much time looking at these different things right now, but you can see how these different things relate based on how these things are and where they fit in the chart. So again, if you're interested, you can spend more time. You can pause the video and look at these things. But for now, I'm just showing you this as a classification technique. Okay. So the last thing we're going to discuss is, again, looking at the assigning of scientific names. We call this binomial nomenclature, meaning two names. You have a first and last name, and so do all organisms. Those organisms have what are called the genus, which is the first name, and the species, which is the second name. Okay, so the genus is capitalized with the big letter, and the species is lowercase. Okay, so when you write things out, you have the two names, the genus and species. You always write it out, or uppercase or capitalize the first, the genus, and lowercase of the species. Okay. The other thing you always do is that you either underline it or you italicize it. So we do this to identify scientific names. So you either underline these things or you do italicus. Okay, so that's what you do here. You look at this, and this is the underline or italicus, and you see the abbreviation, the S. aureus. S. aureus is, again, Staphylococcus. So sometimes once you see it over and over again, we just abbreviate this because this gets long and arduous and kind of tough to always say all the time. So when we get lazy, we just put the S as the genus and then use the species name around here. Okay, again, the inspiration for names is all over the place. And so for Staphylococcus, this is basically what it looks like. It's basically a round bacteria, or coccus bacteria, that has the staph, which is arranged in clusters, and it's kind of gold in color, so that's where it gets the names. But sometimes they're named after scientists who discover them, or a lot of different things. And we're going to see a lot of different microbes as we go along. The thing I want you to get out of here is the genus and the species. First name, second name, genus and species. Always capitalize genus, always lowercase the species, and you either underline or italicize based on if you're either typing it or you're writing it out. Okay? All right, so we've come to the end. I know you're excited. You probably are just like, God, he never shuts up. But these are good videos, so I think you'll get a kick out of these things as we go along. Okay, so we looked at microbiology. It's a study of life. Again, these are study of life smaller than what we can see with the naked eye. And again, these include a lot of different things, including viruses, bacteria, protists, fungi, and worms. Again, we're going to spend most of our time this semester looking at bacteria, but we will talk about viruses in Chapter 6 and some of these other guys in Chapter 5. Okay, we talked about some of the applications that we use for bacteria and these different things. And again, obviously the reason why you're taking this class is due to the infections and things like this. We did a little history tour of microbiology. We first talked about the first microscope with Antony van Leeuwenhoek, and then again we talked about the germ theory and that microorganisms and not a bad personality or bad doing bad things or what caused you to have disease, but it's actually due to the microorganisms. And again, two main figures that I want you to know about, again, I'll ask you questions on the test, are Louie Pasteur. Think of pasteurization, Pasteur in this case, and Robert Koch. He has the Koch postulates. He's the one that does step-by-step thinking about the different diseases and how you do identify a disease based on taking a sample from a patient, infecting another organism, and then isolating it out, getting the same organism again. And that's the Koch's postulates. Finally, classifying microorganisms, we have the three domains. The three domains are eukarya, which are the eukaryotic cells, the bacteria and archaea, these are the prokaryotes, and again, we're going to spend some time this semester looking at these different things, and again, we'll talk more about these in Chapter 4. These we'll talk about in Chapter 5. And then finally, first and last name, know your genus and species. If you have a first name, that's the genus, the species is the last name, and again, genus is always capitalized, species is always lowercase, and you either underline or italicize when you're writing it out. Okay, so that should come to the end of this video. If you have any questions, please feel free to email me or ask me when we meet for lab. I'm happy to answer them for you at any time. I thank you for watching, and I'll see you next time.